The topic is genetics, and genetics can mean a lot of different, it holds a lot of different meanings, and hopefully we can start to uncover some of the challenges that we're facing in terms of genetics, especially single-cell genetics. I don't know if it's, I know you guys probably don't want to move in the back, but if I can ask you guys to come maybe forward, I think we could have much more engaging conversations. So we'll really appreciate that if you could come a little bit forward. So my name is Jay Shin, and I'm from RIKEN, and I'll have Saihan introduce herself.
Yeah, I'm Saihan from Genome Institute in Sydney, Australia.
Great. So you heard her talk yesterday. Fantastic work on pulling a lot of PBMCs and doing single-cell genetics, hopefully through the EQTL. I'm part of the AIDA project that Shyam mentioned, so also an attempt to profile many single cells in Asian population. And ultimately, we came up with some questions here that we would like to dive into, but I think the reason why we have this discussion is this topic that was mentioned by Kirsten Adderley yesterday and Shyam, that only 1.1% of GTEx data is from Asia. And you could see this in many other genomic consortiums that Asian genome reference is highly, highly underrepresented. And that has a very severe implications downstream in even interpretation of SNPs and developing drugs and so on. So how can we address this point earlier on in the consortium and really motivate the community locally here in the region to generate the data as much as possible? So let's just get into this. Again, there are probably a lot of things that we can do with the genetics information, but what will be the main purpose of having and generating single-cell genetics data points? So I want this discussion to be you guys talking more, because I'm not an expert in genetics, but I'm sure many people here are. So what will be the real benefit, and you know, what can we really truly gain through having a single-cell genetic information broadly, but also in more specifically in the region in Asia? Anyone want to chime in? Chung.
Thanks, Jay, for the opening. I think we should separate the diversity and genetics. So two things is slightly different. You could sample all the populations without doing genetics. Okay, you could achieve the equity without doing genetics at some point, right? So genetics, I assume we are talking about something like EQTL-based study, right? So, and technically two things are separated.
Okay.
Yes.
I mean, when we say like in the genetic studies, what do you, so first question is, how many of you are actually working with the like genetics data other than like, you know, DNA sequencing, let's say, or genotyping? 1, 2, so a few people are so familiar with like the data type, like what other studies has been done. So yes, so I think diversity and equity is like another issue like that we should be addressed. But I think the first thing is like, when we say genetic studies, like what are we meaning by that? Are we meaning population studies? Are we meaning like, you know, functional studies that are going to contribute to your ongoing projects? So I think we need to like differentiate that, and those are two different things to, or they are going to get like different, they have different aspects like to discuss. So both is required, I think, for the like, you know, the long term to answer like health questions and disease questions. But how the community wants to move forward about this.
So maybe related to that, and yesterday we talked a lot about this, is what kind of questions you guys have? What could be the question that we can ask with this data set, or prior to generating this data set? What are some of the questions that you're curious about? Biological questions.
Besides EQTLs?
Besides EQTLs. Is the EQTL the only thing?
So I think the QTL concept, that the eQTL just for the expression of genes, but of course if you move to single cell, you have way more QTLs that you could test, and probably that's why people move to single cell. If you do IPS differentiation, for example, you have differentiation QTL, right? So cell type composition QTLs. But the problem is, how could these problems be technically solved with relatively limited number of samples? So not everybody has 1,000 samples as you do in most of the cases. Okay, so 100 samples. It's facing the same problem as this, like trans QTLs, that you don't have enough, statistical power. But whether the number of cells, like per individuals, that offer more data point in some perspective. Okay, so could that be harnessed to sort of, alleviate the problem of not having sample number? So that's something that maybe we could think about.
I think that if we talk about, it is probably the right place to start to talk about the sample numbers, because if a number of hundreds is really needed, I think this is the right place to really say that we need to scale up the number of samples that we will analyze. If it is needed, we need to set up a request to the funding agency, not on the realistic, but to say what is needed and what we will solve with that.
Exactly. So I think number is, yes, we need to come up with that.
What we will solve with that.
But I think I want to talk about what we want to solve with that, to actually talk to the funders, because they want to have the question.
So I think there's a really fundamental biological question to ask by including lots of different genetic backgrounds, and that is, what does it take to make a cell? What is the total space, transcriptional space, that a human cell can occupy? And you can't do that with a limited genetics. And if you take it a little bit further and start to look at specialised functions of cells and asking how different individuals acquire that function under different constraints, different genetic backgrounds, different environmental backgrounds, you start sampling lots of different modalities of networks that you wouldn't see if you had a limited genetic or a limited environmental profile. And that's exciting, because then we can start to dissect out not just what makes a cell from a developmental perspective, but you can start to think about the engineering principles behind that, mathematical principles behind that, that might allow you to construct some of those functions in models like pluripotent stem cells. So there is a much broader context than simply understanding human genetics. This concept of what is a cell, which we were laughing about yesterday, is a question that comes up at almost any Cell Atlas meeting, can only be addressed if we include diversity in our experimental design.
Agree very much with the point that, I guess when you say what is cell, you mean what is the cell type?
Well, no, I actually think we're asking what does it take to make a cell a human cell? What are the transcriptional limits of that human cell? And then how does that cell specialise? And I don't think that necessarily relies on cell type. I think you can start to come down to cell function, which might be independent of cell type. It's a very nuanced distinction. I fully appreciate that.
So, and also I think you brought up the question of the development and environment and so on. So maybe we should just define the parameters of this discussion, because nominally this is a single cell genetics discussion. Doesn't mean we have to limit it to that in a very rigid way. Do we want to expand the discussion to say simultaneously looking at genetics and environment or something like that?
Yeah, that's actually one of the last questions. But
okay.
yeah, absolutely. We can talk about that in conjunction with genetics, because those genetics, they don't explain everything about the disease, unfortunately. I mean, most clinicians or geneticists will tell you genetics may explain 10, 20% of the phenotype you see. So what are some other confounding factors that we can work with, and also take advantage of the region here. Environmental is a perfect example where in Korea you eat kimchi, in Japan you eat natto. These are like highly fermented food. How does that impact micro gut biome and thus your immune system? What are some of the factors that we can contribute in addition to genetics? Would that be something that's interesting? I see a lot of nods, but how can we go about that? Or which cell type should we go for?
Right. I think I fully agree. Genes and environment, genetics and environment together is a logical goal and something we should study simultaneously. And certainly in IDA, we want to look at genetics and environment together.
So also, like Jimmy Yee's lab in UCSF is doing Asian populations in the US,
right,
on PBMCs. So we do need to probably harmonize with them very nicely, which we'll get into later, which can probably address both genetics and environmental factors.
I mean, I think if you're doing genuine population surveys across Asia or any part of the world, that it's very hard to deconvolute genetics and environment, particularly if you're working in very highly polluted cities, cities in low to middle income countries that may have different sanitation standards, or environments which actually promote longevity, just as Piero was talking about earlier this morning. It's hard to detangle.
Yeah, so I think that's really the scientific question we have to address. So, for example, we're trying to look at the Indian population in Sri Lanka, India, and Singapore, or Chinese people and Chinese Singaporeans or Chinese origin Singaporeans versus Chinese origin Chinese people or Chinese people in other countries. So I think that's a first step. Like you said, there will still be confounding of genetics and environment. But that is really the question I think we need to address. I want to add one more thing on what is the purpose of doing single-cell genetics. QTLs certainly are the obvious thing. I think differences between population groups is very important, regardless of whether we figure out why those differences exist. It's very important simply to say how are Koreans different from Japanese, different from Singaporean, different from Chinese, and Thais, Australians, Malays, anything. Because that has impacts on everything, impacts on diagnostics, impacts on disease risk, impacts on mechanism, precision medicine. And then, of course, the moment you define it, then you'll start saying, well, why does that difference exist? Is it genes? Is it environment? But I think it is a very important objective of doing single-cell genetics.
So just representation to make a better reference, right?
Okay, that's the third thing. What I was talking about more was how are Japanese different from Koreans?
Oh, okay.
That kind of question.
I see.
Right.
In terms of genetics or
Molecular traits. Molecular traits.
Molecular genetics.
Yeah. No. Let's say gene expression.
Gene expression.
Right. Yeah.
I think the purpose of doing single-cell genetics, there's another aspect that is not individual-wise, it's single-cell-wise. So there has been reports that somatic mutations are probably have a higher frequency than we expect. So for a single-cell for HCA study, we should be more interested in those somatic mutations within each individual, whatever genetic background he or she is from.
So somatic mutations in the exon regions?
Yeah, whatever. So, and their impact on the, say, transcriptome.
Yeah.
Technology seems not so mature yet. So there are single-cell genomics, but the data are quite sparse and not covered. But someday it will.
Well, yeah, so Stefan Quake showed in one of the single-cell meetings where in aging mice, the mutational load is increasing by just using full-length single-cell RNA sequencing. you can measure these mutation rates in the exons of the transcript. And as you age, these are more frequently observed. But so yeah, we'll get into the technology of how to do all this, but at least if you want to look at exon mutations, we can consider full-length sequencing.
So there is not only a genetics of individuals, but also a genetics of cells within a single individual.
Yeah. Okay, I know that, what is the current, your opinion and also the mood of the community when it comes to GWAS? So, GWAS has been around for a long time, and one of the main missions and purpose of doing GWAS is to identify causal genes which can be derived to target for therapeutics and applications, and we'll be all cured. I mean, that was the promise that the Human Genome Project was really laying out for the future, but reality is that it's much more complex than that. So now there are, of course, integration with EQTL, because many of the SNPs are found in the non-coding regions, and how do they regulate gene expression? Do you think that would eventually help us to solve the problem? Or do you think, kind of going back to this question of purpose, are we doing this to what end? To develop drugs, or for therapeutics, or precision medicine, of course? So what are your impressions thus far from your experience and knowledge with GWAS studies, and how do you see it going forward? And how do you think single-cell genetics can actually make it better and improve this?
So I think that's a very general question, but if you look at how people uses GTEx data for whatever they were doing in explaining the GWAS signal, that almost everyone would be pretty much certain that if you have high resolution, broader scope of a GTEx type of data, that would definitely benefit the field of genetics, for sure, 100%. Okay, so, well, I mean, the easiest way of thinking about it is for a project like GTEx is tissue-based, okay, so, and we all knew, right, so you have specific cell type in there that contribute, to that particular specificity. So, and the increase of resolution, that what I would say for now, that the baseline of what we are doing with the single cell type of EQTL is, in most of the cases, the increase of the resolution of what the GTEx has been done, right? So, and that contribute to certain values, for sure, okay? So, and whether in this paradigm that you could have something more than just trans EQTL, more than just cis EQTL, more than just splicing EQTL. You have another type of QTL that could be used for interpretation for more than just gene expression. Okay, so that would be another layer of benefit. Okay, so but this, for now, yes, nobody knows that what is the resolution and what is the power in these types of EQTLs.
So I completely agree with your points, and having single cell resolution will be much better than what has been done now. But I guess maybe I'll rephrase the question again, taking a step back. Having all of that data in the end, let's say we have perfect data of single cell genetic information, how will it be helpful or beneficial to the society as a whole? I mean, that's kind of do you think it's going to be much better than GWAS and current EQTL understanding? And even if it will, like what benefit, what applications can we consider? And I'm saying this so that we can all go home and tell our funders the translational benefit of genetics.
Can I brainstorm a little bit? And
of course.
Many things that I will say will not be realized at the end of the day. It's all recorded, okay? So, but I think that clearly with EQTL and GWAS, we will all going into gene dosage and understanding what regulates the gene expression. So in my conflict of interest part of my. So I have a small company that is focusing on haploinsufficiencies by restoring expression, particularly in some genes that has to be expressed in the brain, plus minus something, and if properly expressed, everything goes well. Half copy is very bad. If you overexpress, is toxic, and all the cells die. And and clearly, I think that all of this will tell exactly what happens in each cell, will give us the information for future therapies, when we will figure out how to switch partially one enhancers or regulator to the right level, to the safe level, to the appropriate level. So we'll figure out at a certain point how to activate some specific genomic region. And it's very important, cell specificity is absolutely important to express things in the proper way. So I think, so particularly when we are brainstorming with my other collaborators, are we going to target a gene is haploinsufficient in a certain cell? We really need to know. We really need to know exactly in which cell it is expressed to really be sure that we don't damage anything else. Certain diseases are very delicate and really want to be accurate. So clearly, what is missing so far? I mean, I believe that we can start to study what regulates gene expression in each cell. So once we know the expression, shall we develop CRISPR type, or shall we? Will we identify very specifically expressed non-coding RNAs that are important for the epigenome function in a given cell? So I think but clearly, a catalog of expression at a very high resolution in every cell is very important for the following studies to happen. And it's not going to be something we can tell to the funding agency. In 3 years or 5 years, we're going to have drugs. This is a path in the long way.
So you mentioned haploid insufficiencies. So do you think we should do single-cell allele-specific expressions? How do we achieve this in human? Because that's related to the genetics.
So I think allele-specific expression is a very common kind of analysis in, alongside EQTL analysis. I feel there's no reason we can't do it in single-cell data. But I would lump that under the EQTL broad category. I think your question is a very good one. Why are we doing this? Maybe the answer is, in a way, standard. It's like, why do people do EQTLs? There's two things. One is understanding what's the parts list of our genome, what is the mechanism, what are the functional elements in our genome, and if you have an EQTL and you fine map it to this regulatory element, then you know that this regulatory element regulates that gene. And that is just a very important endeavor to understand which regulatory element regulates which gene. Obviously, you'll only be able to do this for the regulatory elements that have meaningful mutations, but that's okay. That's still a very important step. So I would say just basic biology is very, very important, this kind of catalog. And then comes the GWAS part, which is you have a GWAS variant. For common diseases, they're mostly non-coding. And they essentially exert their effect by affecting some gene or some transcript. And so EQTLs are the main way of getting to that, right? Understanding the mechanism underlying a GWAS locus. And that makes that GWAS locus, that GWAS result a bit more credible. And then you understand the mechanism of that, then you can start saying, okay, I understand something about the disease now. And I understand what is the role of this gene in the disease, what are the upstream factors that regulate it, and so on. So there again, we're talking about disease mechanism, and solidifying what we think we know from GWAS, right? So I think that's just an incredibly important effort. Just having a GWAS locus without knowing its mechanism doesn't help as much if you want to design drugs or perhaps even diagnostics.
Being able to connect the dots that GWAS, but now you know you can make these two locations association studies.
Right, and which is the causal variant?
Correct, yes.
You want to know the causal variant, right?
Look, I'd strongly like to echo those sentiments and further add that we actually don't know what all of the genes in the genome do. So this cataloguing is extraordinarily important. In fact, for a large number of genes in the genome, we're still guessing through ontology-based annotations. Now, being able to overlay very discrete cell type expressions and co-expression networks of molecules that exist within the same cell is a really important part of building up that functional annotation of those genes. And even more important for genes that lack ontologies based on protein coding, so the non-coding component, of which we're really quite blind to at the moment. But coming back to your question about G1, I wanted to ask you a question, Jay. Are you suggesting that as part of this consortium, we should be collecting the kind of phenotypic information that would be needed to conduct additional genome-wide association studies across this data set?
I mean, that's a big question, and yeah, you want to say something. What do you guys think? Do you think it's necessary? Should we collect phenotypes?
It's an opportunity to
Exactly, yeah.
Yeah, exactly. So I wasn't all the worry why we wouldn't.
In either we're collecting as many phenotypes as we can, and we have a core list of phenotypes we're collecting across all 3 sites as minimal standards, and then each site has its own special interests that they add over and above. But I mean, it's an excellent point. It would be, as Piero said, a huge shame not to enrich that data set with various other measurements.
So yeah, right now it's just focusing on healthy individuals, but maybe once we get into more diseased individuals.
Yeah, so something as simple as BMI or blood count, things like that. How do the molecular gene expression values you're collecting and the genetics you're collecting, how do they correlate with all of that? Are they causal for these things you're measuring, like blood count or cholesterol? Or does cholesterol influence gene expression? All of that, I think it's very, very important. So I would say a genetic study in this space, it has to genotype the individuals, it has to measure their gene expression or gene regulation, it has to measure covariates and any phenotypes that we can, it has to characterize the environment as precisely as possible, and then we'll really understand all of the determinants of why our cells are behaving a certain way.
I would like to raise a point and would be curious of what people in the room think about it, which is that of all the cells in the body, B cells and T cells are special because they are different in every person, and they are also different in every cell, right, because of the VDJ recombination and somatic hypermutation in case of B cells. And when you go to single cell, people have done immune repertoire sequencing across populations, et cetera, and obviously Japanese people have different HLAs and repertoires from European people, et cetera. But the single cell brings a special kind of ingredient to the soup, which is that if you do single cell sequencing of those things, then you can clone out the antibody or express recombinantly the TCR, and then that's going to work and target the exact same antigen that it did in that person. So the single cell really brings you a very translational kind of ingredient, which is that suddenly it's not only, oh, I know the sequences and the HLAs, but you can actually make an immunotherapy based on the antibody or something like that. So it seems like profiling kind of diverse populations, et cetera, is important for this kind of environmental causal relationship, but also especially for immune cells because of the potential for discovering good antibodies or good T cells out there. I don't know what you guys think about this specific topic.
An excellent point, yeah. I think we all agree.
Are you guys doing it in IDA, for instance? I was wondering whether you guys do TCR and PCR sequencing for this purpose.
So we're doing the 5' based approach in IDA, so we can take both the promoter enhancer information, but at the same time do TCR, PCR. Yes.
And do you decide what people you are gonna spend your money on based on their HLA types to diversify as much as possible, or is that not a concern, or you just sequence everybody until you run out of money? Or?
At the moment, yes. But you're right. Maybe in the second phase, you can selectively choose individuals. Could be of interest. Yeah.
I was wondering if you had also follow-up check on these samples that you collect and to see if in future they got some kind of health problems. Then you have some data which also is gene expression and also the genetic variations, and then you could make a better sense of those things and associate it with particular phenotype like disease or susceptibility to infections, and also with having this clonal expansion information. I guess it makes most sense for analyzing the data in future if you have some follow-up plans for your samples. Yeah, I don't know.
Yeah.
Help,
Well, so that's an excellent point as well. So, of course, the time of acquisition of the samples, that individual is healthy, but in the years to come, that person can develop particular disease. And so just tracking that individual, which then becomes a whole ethical issue later on. But it's actually a good point to consider preemptively when you are writing ethical documentations that you would like to do follow-up on these individuals for another maybe collection of the blood or just their health checkup information, for example. Yeah, it's an excellent point. Thanks.
So I just want to say a few words, because my background is in actually population genetics. So I've been involved with lots of genome-wide association studies. So we learned a lot from those studies. So there's a huge. This is not a simple. The endpoints that we want to reach are basically in all of the I think the medical science is what we want to do is we want to learn more about how things are functioning in the body. And the second thing that we want to, can we identify actually those who are susceptible to disease? So we can actually prevent, maybe manage the disease better. And the ultimate aim, of course, to cure. But we are not going to be able to do this by ourselves. So we are a community that who have expertise in single cell genomics and this understanding. So I think we need to look closely to what has been previously done and what learned from those studies. And with the population genetics, I can say that GWAS studies, there are, of course, when we looked at the first GWAS study was actually published in 2006, but it's now today we have very powerful GWAS studies because people are coming together now. And the study methodologies also developed over the years. It wasn't until a couple of years ago that we agreed on the methods to actually do genome-wide association studies. So there are different aspects of this problem that we need to address at the single cell level. And the other thing that I wanted to say is that we can learn all from these, and we can start those collaborations now. And so one thing with the genome-wide association studies, I heard this a lot of conversations. Oh, do we need large sample sizes? Do we need small sample sizes? So this actually depends on the question that you want to answer. And if you look at the first genome-wide association studies that has been published on age-related macular degeneration, the total sample size was around 256. And from that study, what they have found is actually the VEGF receptor, which is actually then taken by the pharmaceutical companies, and now we have the anti-VEGF treatment to treat age-related macular degeneration. It doesn't cure the disease, but it actually lessens the progression of the disease. So that was a huge, and since then, this was the motivation why genome-wide studies has been very popular. But when we look at other diseases like diabetes, or because my background is in eye genetics, is or more complex diseases such as glaucoma or short-sightedness, we get together with sample sizes of 160,000 individuals, and we still get only 10% of the genetic variation that's explained. So I think it really depends on the question that you are trying to, or the disease that you are trying to know more about, that you have to design your studies. Also, with this about how we're going to do phenotyping is if the data, if it doesn't, so phenotyping will give you better understanding of your data, as we've seen in Kristen's presentation yesterday. So by looking at your data, you're going to get some finding, but getting to nitty-gritty things of things and to understand and deconvolute all those factors, you need those phenotyping. And this is exactly the same thing we have done in the GWAS studies. So if you have large enough numbers, and if you just ask people, do you have this disease or not? And that person may not actually know whether they have the disease or not properly, or they might mix it with something else. And that's what we see in the UK biobank studies. The people were asked, whether they have diabetes, or yes, some of them has type 1 diabetes, some of them has type 2. So they are two different, but people are not going to give you the right answer. But that data is still gives you some understanding or some results, because it's the sheer number of people you include. But there is this opportunity that if you phenotype really well, then that gives you another level of information. So I think you need to, since it is harder to do, I think, large-scale studies in the single cell at the moment with the cost, it is better to focus to phenotyping, and then eventually, of course, we will move on to doing larger scale studies. That's my perspective. And what we learn from the population studies. And so at the moment, how we are doing these GWAS studies is we have consortiums. And in the consortiums, well, I'm an analyst in 2 of them. And so how we are running the analysis is we ask everyone to give us the metadata, how they collected their data, what measurements that they have done, and you're asking for certain covariates. And then we write our coding. So one of the groups leads the analysis, and we write our coding, and then we send out that coding to all the analysts in different studies, and then they run, and then we collected the summary results and do meta-analysis, and then that means that making sure that we are all replicating what everyone else is doing. And of course, different populations are going to be different. And that's the case. When we are analyzing the meta-analysis, we keep European populations together, and then the East Asian populations together. And it has been very fruitful for this type of studies. So I'm telling these for you to to think about it, that how this community can do something similar to that, and perhaps create a framework for if you are actually interested in studying the same diseases. But the question that you are trying to answer should be, I think, somewhat similar for for everyone to work together.
That's an excellent point. And so I guess this framework that you've mentioned, so how can we go about this? Do you think within HCA Asia, could such framework exist?
I mean, as I said, it really depends on the question or the disease focus. We know, like, and we have to remember that complex diseases is like totally different to cancer. I know, like, that lots of people went the other side. So cancer genetics is like a really important, and like, you know, in the complex disease, genetics is important. And there's this issue, of course, with the funders, like what they're gonna, like, you know, do. So can I ask a question that how many of you are actually working in complex diseases? And how many of you cancer genetics, cancer biology?
They're over there.
Okay, all right. So why we have this in this room? Okay, so I think it will be good to like have this discussion of like, you know, to first bring the disease focus groups. That will be my approach. I guess. And even like, you know, if the diseases are different, that's okay. If we are like, you know, somebody's like, you know, studying inflammatory diseases and like the other person is like working on the Crohn's disease or I don't know, but like any other like in complex disease that you can think of, then like, you know, to learn from each other, what type of data is like, you know, collected and I think the biggest, like, you know, thing is, like, as we seen like from the GTEx, is like, you know, tissue collection. And we need to phenotype that pretty well to be able to remove, like, you know, any effects that we're going to see in the downstream analysis, including the EQTL analysis or any other functional studies. So I think maybe, like, you know, coming up with a single cell, like, data framework, how to collect tissues, how to preserve them, but the only thing that you have to always remember that this is a very new area and there's so much unknowns. We still like having discussions about how we're going to preserve the tissues to like, you know, use them in the long term. But this is, I think, good opportunity to start these discussions and maybe to discuss what we are going to include in these frameworks.
Yeah, thank you. Yeah, so I mean, yesterday we heard a great talk from Christine Ardley, and that was very much kind of on-site, single institution taking charge of the whole effort of multiple individuals. Now, Human Cell Atlas, by nature, is very different because we're much more decentralized, and you may be doing your own particular sample preparation. So it gets into how we can harmonize these efforts and come to a way of standardization. So I mean, Shyam, Unyang, myself, Chung here, we have teleconferences to how do we prepare PBMCs? And, you know, that's also such a simple thing as blood. It could come into a lot of difficult issues because do you use heparin or coated, or do you use EDTA coated syringes? To the whole questionnaires that you're going to be asking. How do you come up with this consensus questionnaires that we can use to compare our data with? So it's a lot of effort, but it's worth it because we get to harmonize and collect the data from different sites, and it just synergizes the data. So how can we expand this to not just PBMCs? If you're doing PBMCs, in your lab anyhow, I think it'll be great for one of us to then share the protocol so that anyone can follow in the same way and can harmonize the data that way. But if you're doing a particular tissue, how do we achieve this in a regional wide or global manner? It's a challenging question. But.
You probably need to organize some courses or demonstration where everybody interested fly to a given place, and perhaps the Asian office may, further grow. The Human Cell Atlas may further grow to start to coordinate this kind of activity as well. Maybe we can add this as one more thing to do for the office.
Yeah, so like an overseeing office to help people bring together. Yeah, so I think that one of the unique challenges of Asia compared to, say, Europe or the US is that U.S. has a central funding agency, NIH, and they can actually bring a lot of people together through just funding the right people, right? Creating that network. Europe, you have the EU, but unfortunately in Asia, oh, Jonah's there, thanks.
This is a typical thing that
Yeah, I mean,
things that cannot be done by any funding agency, right?
well, I mean, that's kind of the thing, is we're trying to increase the genetic diversity and representation, and one of the reasons why a lot of the Asian populations were underrepresented in many of genome studies is because of the funding. And the fact that the funding is also very much decentralized, where there's no central governing body within Asia to say, this is the common project that as an Asian country we're going to go. We lack this. Unless, like, there's an APAC, which has completely different economical purposes, but for science reasons, that's one of the big, I think, personally, it's one of the reasons why there's so hard to coordinate these big projects. So I think it's a good idea to have a coordination in terms of bringing people together, but ultimately, how can we, again, it's a very difficult question, how can we kind of pile the funding together so that it will actually synergize towards a common goal?
Maybe this is a sort of a side comment. So what strikes me was when the GTEC talk yesterday, talking about sample collection and how if you've locked in a sample collection to a certain way, that may exclude other omics potential. So single cell, what we are discussing here, in some sense, is one type of platform. There's still many different type of platform, and today we see all these multi-omics. So this discussion actually is happening here, but at the same time is happening at the mass spec space, it's happening at different space. It would be interesting, I don't know whether there is a way to potentially engage with other people doing the same thing but on a different platform so that we synergize effort. Currently at Senior Australia, looking at metabolomics disease, we spend a lot of time discussing the collection of blood, fasting, non-fasting, the time. That's a huge sample collection protocol so that whatever we collect can be utilized for SNP, transcriptomics, lipidomics, metabolomics, microbiome. So we could do that. And I think maybe we should see this so that the effort won't be wasted. Getting to the person is hard.
Yeah, thank you for bringing that up. At least in Singapore, we're trying to do exactly what you suggested. We're trying to piggyback off the national precision medicine cohorts rather than starting our own. And so thankfully, we got a very good response from them. And actually, some of them are already our co-investigators in AIDA. So it makes a lot of sense because it's not just that they're collecting samples, they're also collecting a lot of metadata. That's right. And they're doing whole genome sequencing. So it's a win-win, actually.
So yeah, it's a good point to consider multi-omics in advance and prepare the samples in a way that for the future. I mean, so that's kind of why I asked the question yesterday to Christine is, what will be the one thing that you would do better next time is that she'll freeze all the samples, for example. So perhaps maybe you can consider if you are doing any tissue resections or biopsies to make sure before you dissociate them, have some part frozen down or in sections so that you can maybe repurpose them later in the future. But how many of you are actually doing sample preparation in this crowd? So not so many. How many of you are like computational genetic? Yeah, most of you. Okay, so you just want the data. So yeah, I guess really how we can harmonize protocols. Should we have So right now we've been kind of talking very broadly about, different disease, but should we have a topic or discussion about particular disease or samples or tissues that may also be relevant in the region? For, again, we're in the HCA-Asia meeting. Are there particular cancer or disease that may be more relevant for this region that may also help communicate the benefits of this?
So I will now plug in my research here for a minute, but I study flaviviruses like dengue, Zika, and all these kind of things, and other RNA viruses that are very common in tropical countries like Indonesia and Vietnam, et cetera, and they are virtually absent from Europe, and they are mostly absent from the US, except Hawaii and south of Texas, etc. So all this kind of infectious viruses stuff is a lot more relevant in Asia than it is in both Europe and the US, and it does relate to genetics, right? Like it's clear that depending on your background, you have a better immune system or not, and so on. So, I mean, I study infectious disease, so I'm interested in that, but I think that that may be an interesting thing, that all these tropical diseases are much more relevant in Asia than they are in these other areas.
Jung?
Thank you. I think that if we talk about like sampling from different regions, and I think for genetic studies, the way of sampling is very different from like a regular, single cell project that you take 5 or 10, 10 versus 10 you get all your conclusion. But all these genetics we are talking about, at least 50, maybe better 100, 200 is better, right? So in this type of sampling, it's not any regular sampling you could do in ordinary lab. Especially, you could do it with blood, you could do it with skin, but with lung, with liver, right? So this is, all of these sampling is done by a lab that has a very specialized expertise on that particular tissue. They have access to at least 50 to 100 of those. Okay, so, and this kind of sampling has to be done by forming some sort of seed network across the country. So you have the lung lab in Japan, the lung lab in China, the lung lab in Korea. Everybody give 50 lungs, then you could have some of these transethnic genetic studies. Okay, so unless that we are not talking about genetic study, just talking about get 10 Chinese, 10 Korean, okay, and 10 Japanese, and look at the transcriptomic differences, but no genetic differences. Okay, so then it's a different level of sampling. I think we have to think about it more systematically.
And that's exactly the challenge, right? Is that probably there are some also language barriers and cultural barriers and funding barriers that really, there's geographical separations also. But how accessible is it in your community, in your country, in your university, to actually acquire tissue that you're interested in? Tissue banking, is that readily accessible, or can you how easy is it to collaborate with clinicians to acquire these samples? Because Goji is not here, but he can access any tissues. Like in Japan, it's also very difficult to get healthy tissues. So the concept of like organ donor is not as widely pervasive as in the US. The whole postmortem sections are also very challenging and limiting due to either cultural or religious reasons and historic family reasons. So is that similar problems you face in your countries?
Singapore does not have a national tissue bank. And so it really depends on individual clinicians and individual investigators having access to certain things. And they tend to be disease samples. So like you said, getting healthy samples, I mean, there are organ donor programs in Singapore. So it's a matter of tying up with them. But it's serious work. It would be much easier if a tissue bank just existed or that we could access. And we work with them to say, preserve the tissues this way for our assays and so on. So to be frank, over here, that's why PBMCs are a great and easier place to start for us. We can do cancer also because the tumors are removed in routine clinical care. Beyond that, getting healthy tissues is not straightforward here, I would say. A lot of legwork to set up the ethics, build a relationship with the relevant clinicians. So I just want to know, is anyone in Singapore or outside Singapore, how do they access healthy tissues for this kind of study?
So hi, I'm Amelia and I work with Prof. Tai Yishong, and we do have a collection of healthy skeletal muscle transcriptome, and I believe we have some frozen tissues in the Chinese, Malay and in Asian Indians in Singapore. So I would say, I think there are studies in Singapore that look at healthy samples, but for me, we're looking at pre-diabetic. So they're coming into the diabetes clinic and they're obese, so they aren't exactly healthy, but still pre-diabetic. So I understand there's also the Gusto study that looks at the longitudinal outcome. So I think they do collect maternal samples before birth and then they follow through. So I think there are some big studies that collects tissues across Singapore. But yeah, you need to approach Gusto and Singapore Adult Metabolism Study. And I think there's an Asian study coming up, which is a longitudinal study looking at diabetes progression. So those are the complex diseases that are actually very heavily looked at in Singapore, especially diabetes. So Prof. Tai, I looked at the skeletal muscle transcriptome and I looked at the genome and I did the EQTLs and the GWAS integration and so on. Yeah. Gusto, I think they do have some placenta samples. Yeah. I'm not sure. I think for the children themselves, probably just blood.
Yeah, I think I'm aware of the placenta collection, and I may have heard about this skeletal muscle also, may have talked to Ishiong about it. So it'd be great, at least in Singapore, to do some kind of census of what tissues are available. But I think Christine had a...
Look, I realise that the Human Cell Atlas is about building a healthy reference, but if we just turn it on its head a little bit and come back to this question of what is a human cell and what is all of the possible phenotypes that a human cell can exist in, then by excluding disease, you're excluding a very wide part of that human existence. And the lessons that we will learn by including cells which acquire phenotypes as a result of disease, somatic mutation, environmental exposure, even just ageing, means that we are building a very narrow view of all of the molecular networks that a human cell can support or that drive particular functions. So if we look at disease as an opportunity to build in those perturbation networks or extreme networks that we wouldn't see otherwise, we're going to get a much more informative map of human cell biology by including the disease paradigm. So I wonder, is the Human Cell Atlas Asia the opportunity to do that, given the funding pressures and the sample collection pressures? And is this the value add that it can include to the wider Human Cell Atlas project?
Well, maybe I can comment. So, well, I think they're absolutely right. There's multi-layered answer to this, and I think as a global Human Cell Atlas, correct me if I'm wrong from the DCP team, but I think there's no restriction as to what kind of data you can upload. Anyone can upload their data, or does it have to be healthy tissues or not? Or so if if I have 10 cancer cells and 10 healthy cells, you would take that, okay. But like to run the pipeline and to consider the reference, we will not consider cancer tissues as a reference, but it will later be useful to compare, right? And in a way, in Japan also, we are creating this network to acquire tissue samples, and a lot of these clinicians or PIs want to keep their disease samples in a way that they want to be able to publish first, but they're more than happy to share healthy data, for instance. So there are some dynamics that also needs to be resolved in terms of how do we protect the first data release and publication for individual researchers in Japan. But so in that regard, I think there is, and again, just getting healthy tissue is just difficult also in Japan. So we will always get something disease or some unaffected regions and so on. So again, I think we would have to have a good metadata and annotation so that later on when DCP needs to then filter out what should be included and what should not be included for the reference can be easily accounted. But yeah, I don't know how, again, in other countries, what are some of the accessibility to tissues or other tissue banks. Would you be able to perform GTEx, like studies in your countries? Yeah.
Some comments. I think nobody on this planet could do another GTEx, right? So it's 1,000 people, right? The whole body. So, but what you would expect is in certain countries that some of the labs would have access to one particular tissue, right? So I think what we have to do as an Asian consortium is we have to have some of these organizing committee could hook up some of these lung in China, lung in Korea, so we could do some trans-ethnic EQTL with the Asian context. So that would be exceptionally valuable because if you look at almost all the EQTL studies, of course, it's Caucasian-centric, and second, it's not trans-ethnic, right? So if we have to infuse this equity, component into the study, my opinion is we have to do it this way. Okay, so someone has to organize and hook up the lungs in different countries, for example.
I think as excellent reminder for the organization committee of the HCA, I think there is a membership registry with the members which tissues they are interested, but also which tissues they have access to. So maybe perhaps we can go through that list, look at the countries of the region, and maybe like someone in the coordinating office can start creating new networks within Asia. Yeah.
So at the equity meeting in Ethiopia a week or so ago, there was a lot of discussion about the fact that low to middle income countries have limited research resources. It's very difficult to justify exploratory research, and that they need to frame any kind of sample collection around benefit back to their communities. And so excluding disease tissues may actually become an equity issue representing countries across Asia which aren't as wealthy as Singapore and Japan, for example.
Thanks for that comment.
I think that was a very interesting point that was brought up in Addis about the benefit going back to the people contributing samples. And it's something I think would be great to address here in HCA-Asia also. I don't think we finished the tissue access topic, but that's fine. We're running out of time. We can at least touch on this. But everywhere in the world, single-cell analysis is done in wealthy countries relatively, and in the wealthier cities of the wealthy countries. And so it leaves out only certain kinds of people get sampled, and certain kinds of samples get sampled. How do we expand that, firstly, so that we even, let's say we have, Malaysia in the HCA-Asia, but it's not the, high social and economic and educational status Malaysians living in nice parts of KL in Kuala Lumpur who get sampled. It's actually the full genetic diversity and economic diversity of Malaysia that gets sampled, for example, right? And I think that's a very key part of doing any kind of genetics study here. And then, of course, the issue is why should those low economic status people participate, what is the benefit to them? My personal take on that discussion in Addis was that it's not that easy. We first have to do the science, then way down the line some benefit will materialize. Going in saying, give us your samples, you will benefit from the science in this manner, I think it's just hard to come up with that, right? So at least my understanding from talking to epidemiologists who work with LMICs, the low and middle income countries, is that the benefit, the immediate benefit that they can promise at the outset when they start the study is not that your community will benefit from science, but that the researchers in your community and the hospitals, we will train them. So that is a tangible benefit they get from participating in the study. You define what kind of training you want, what kind of technology access you are interested in is relevant for you. We will provide that in return for your participation in the study. So I think training is something we can definitely and should offer in HCA to widen the circle of people who are participating in it. But then the scientific benefit will come like way, way further.
What's your proposal?
So right now it's kind of ad hoc. We're talking to some collaborators and still uncertain, to be honest, what specific training we'll provide. But our tentative idea is to send people from my group there and so do some on-site training, have some people from their side come here to Singapore and be embedded in our lab and learn the methodology here and so on. It was early days for us for sure, but certainly other people in Singapore and elsewhere have been further down this road. And I know Alex Shelek and Musa and all that have done pretty formal training on a much wider scale. I think that's a sensible model. But let me just bring that question up. Is anyone here thinking about expanding the circle of who gets sampled in your respective countries?
Australia is just starting to talk about, to the Indigenous communities in Australia, about whether or not they're interested in being included in this effort. And I think you're right, it's a long-term conversation, and so unlikely that we'll include an Indigenous program early in the Human Cell Atlas days. And not least because we need to mentor and support the Indigenous leads who are going to be driving that effort. But I will say that there was a really interesting point made at Addis, and that is that in many of the low to middle income countries, there is capability and capacity issues, right? So there are scientists who are able to do these studies, but who need additional training or resourcing to be full partners in a project like this. And so I guess the question is to HCA-Asia, is there an interest in going out to those countries more broadly, more proactively to seek partnership, or is this going to be an ad hoc letting them come to you? And that in itself, I think, is an equity issue.
Absolutely, and I think we do need to take more active role rather than passive.
Sorry, I've got sort of somewhat a philosophical higher level point to make that does connect to these things. In any field of human endeavour, you've got a spectrum of ways of organising, from artists painting pictures where there is utter chaos, but lovely pictures come out, to making iPhones, which is close to impossible, but actually we make unbelievably robust, complex things incredibly cheaply. But this is full of rules and discipline and quality control procedures and no freedom of thought at all. So the thing about it is the Human Cell Atlas as a principle is a lovely idea, but actually in nature it's a lovely idea. And the only thing that embodies any kind of rules and structure is the DCP itself. And actually, if you make the DCP too strict and say you're only allowed to put stuff in if it follows all these rules, then basically it would have no cells in it because basically nobody's going to conform to those rules at the moment. And if you look at GTEx, the reason it worked was because it was a command and control infrastructure where we decide what we're going to do, we make the rules, we make the SOPs, everything is followed adherently, strictly, and if it doesn't adhere, it's out. So the problem we've got is actually that we're trying to build infrastructure, which is what the human cell atlas is, but we have no rules. When has humanity ever built infrastructure with no rules? You can't build a road network without rules. You can't build a hospital without building standards. You need rules. And if you look at the only example you can see where there's a nice blending of rules and chaos, which is actually what we sort of want because scientists like chaos because they like to be free to do what they like. And also you get better publication results if you're free to do what you like. You don't get good publication results from building a building, is actually the iPhone. What I mean by that is that the iPhone is unbelievably strict in terms of its quality control procedures to make it possible. But the App Store, if you go to visit a startup app development company, it's utter chaos. It's a pile of smart geeks in a shed creatively making stuff. But if it doesn't conform to the rules of Apple's infrastructure, the app just doesn't work and won't get in. So what it comes down to is where is the balance of the rule sets you have within the DCP. And if you look at GTEx SOPs, they look spectacular. They've worked so hard in such a disciplined way to define SOPs. Why does everybody else go around reinventing the wheel? If there are great SOPs, just copy them. Don't try and invent them again. You know, the US is spending so much money on so many different things. Singapore couldn't spend the same money. Doesn't matter. Singapore's wealthy per capita, but in absolute wealth terms, it's nothing compared to the US. China is very, very, very wealthy as a whole, and therefore China can do things, but copy stuff and define those. And it could well be because of the nature of the community, nobody wants anybody to just tell them what to do. But if you actually load into there SOPs of various kinds and have people vote on them and say, we for the Human Atlas want these to be the rules. If most people vote for those, accept those are the rules, and then nothing that doesn't pass those rules gets in. But at the moment, it just feels like sort of chaos with disappointing number of cells in it. I mean, as in 3 years, thousands of people, everybody wants to use the Human Cell Atlas. Who's truly contributing to it? Because everybody wants their own research results. Well, at the moment, it seems like there aren't any rules. I mean, the DCP might have some rules about what goes in, but they're trying, I can imagine, based on some of the conversations from Wranglers yesterday, was that you're trying to not be too prescriptive, otherwise you reject everybody's stuff. And that would be sad, because then there'd be no cells in. So it's a matter of then, but it's a matter of having the community at large follow rules, which of course people don't like doing, because you like doing what you feel like. And why does this work? Because people don't make money if they don't put the apps on the store. So their funding is directly coupled back to whether they get the app on the store and whether they get people to download it. If your research funding was entirely connected back to CZI saying, for example, you can't have your money unless it goes into the Human Cell Atlas, people would follow the rules. Well, that's good. That's good. That's good.
Yeah, very interesting point. And I guess I've been in several of these meetings, in HCA general meetings, and those kind of questions do come up in coffee breaks and so on. Just my personal observation of this is that HCA is, in a way, unique and very different from other consortiums that I've been to, where ICGC or like Phantom and others are more to have a central funding organization, and they have kind of a very detailed GTEx as well. I mean, you have rules and guidelines of how that money can be used and executed. In a way, HCA is, what I like about it personally is it's very community-based, it's very bottom-up. There's no central funding structure, and that's, I think, one of the reasons why it's so difficult to implement rules is because who have the power or the authority to give them the rule is usually the funding agencies, and HCA doesn't have central fundings. So that is a challenge. You're right, you're absolutely right. We do need some coordination and standardization, but we also need to be motivated in a way, hopefully not just by funding, but through this idea of collection of single cells. And I guess, yeah, I think the time will tell with DCP through collection of many more cells do we see patterns and can we understand the reference? And I think that's just, it's all about timing and the number of cells we need to collect.
Just following up your thing, should HCA as a community have some sort of, maybe it's already have, to go and talk to the funding agency? The thing is, can you entice different funding agency from different countries to push such rule?
I don't know if Jonah wants to comment on that. Question is whether the funders should talk to other funders to create HCA. Well, there are, yeah.
There are already, not in Asia, not coordinated in Asia, but there are already some meeting with the funders and the coordination as well. That's going on.
So, so within HCA, there's an organization committee, but then there's also a group of funders that are, so Jonah is one of them, and Helmsley, and they're mainly philanthropy foundations, but very few, there are few NIH and EU funders, yes. So they're actively involved. Well,
they only require, if they fund someone, for them to deposit in HCA, and your DCP will just increase.
Well, that's, yeah. Can you repeat the question? I think
So you could have philanthropic money, but you could also try to make, I don't know what kind of negotiation needs to be done that for, say, US, NIH or NHMRC in Australia, if they were to fund any funding, we would like to see this information to be deposited in DCP. Just that. It doesn't cost the country anything, but that would increase. Don't know.
Those groups are certainly supportive.
So I guess the point is, yes, yes.
But I think, as Jay pointed out, the Human Cell Atlas, as it's been framed, and as the governance is, it's really scientist-led and independent. And so, I think that this harkens back in many ways to the Genome Project and other initiatives. That I think, it's easy to say that the funders could mandate certain things, but the much more powerful thing is if you, as a scientist, ultimately, make that decision and go through the extra time and effort and diligence required and, kind of define the rules as they were stated. I think that's where the rubber meets the road. So, I think it can go both ways. It could be a carrot or a stick, but I think that the model that the Human Cell Atlas has chosen has hopefully been one that's, the community coming together to chart that path.
Yeah, and I agree. I think loading the data into DCP is one step, but I think we do need to have some SOPs or guidelines or coordinated effort where the data that we will deposit will become actually useful and valuable.
We need some leaders in this group who is going to start this work, who is putting their hands up to do and prepare that SOPs and then share it with the community and put onto a vote. So I think that's the main thing, what I can see from all the conversations. We can go around and talk about this, but we know what to do. We need some rules. And those are for the most important material in these studies is the tissue. So you have to have something that what has the GTEx has already. And since there is so many that kind of documentation available, not only through GTEx, but there is the biobanking communities that's already. So I think somebody just need to do the work and put them together, or there can be a working group within HCA, and so we can start this type of work.
Excellent point.
Thank you. One quick addition, which kind of plays right back into the DCP. So if I have a MD collaborator and he or she wants to do single cell work and has just no clue, I have to do a lot of work that involves explaining them how to dissociate the stuff, how to put their fax antibodies, how to do 10x, blah, 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 blah. Then I have to map their reads, then I have to count their genes, et cetera, and then the fun starts. So if that kind of part of the work is a lot of that can be done by a machine. Like you just put a Wikipedia-like website to tell them, you know, what are the best practices, and they try them out, and then you just map their reads. And, you know, they will pay for the computing time for the mapping, or I can pay, that doesn't matter. But if that kind of work, if that platform is in place to some extent, so some documentation with not super, you know, exotic, but just kind of guidelines, and then also you just do the heavy lifting for them, then they can also read at the end of that page, oh, by the way, this is the standard practices in this field. You have to deposit metadata this way, this way, this way. And then when I go to them and say, hey, we'll do a bioarchive, right? They'll say, oh, that's okay. Otherwise, they'll say, oh, no, you know, in my field, we never published the data before, we never shared the metadata, et cetera, et cetera. So if you give them something that they can take home, they will be much more willing to also then accept what's perceived by them to be community kind of agreed guidelines. Otherwise, there's no way I can go to my collaborating MD and convince them that they have to spend their postdoc time on depositing reads. They'll never do that.
One last thing that I think, I don't know, I would like to get your opinions, like, you know, now we are all scientists, we all understand single cell genomics. And one thing that, like, you know, from my experience working in genetics field, it is really hard to explain what is genetics to a layman person, to funders, to convince them. Even like, you know, clinicians, you might think it's easy to convince them, but as like Fabio said, like, you know, sometimes it takes time, and like you have to prove some documentation. So should we, like, you know, like have some work in this field as well, like, you know, create little, I don't know, but I know from the stem cells, like, you know, there are lots, once it's became like, you know, very widespread, like, you know, area of research, there has been a lot of resources that could, like, you know, explain this in layman terms, so people become, like, you know, familiar with it, and then so that actually increases the visibility of that kind of research, like, in the society, and therefore, like, you know, that the funding bodies and the governments, like, give priority if they, like, you know, see that something is, like, taking off. I think they are, like, you know, more likely to understand and, like, you know, pay attention. So do you think should we have, like, you know, some sort of, like, some resources or, like, you know, little explanations, like what is human cell atlas and why it is important to do it? So, like, I think, like, you know, something like that, not only like for the Asia part, but like also like the greater one. And, like, you know, what's the importance of, like, you know, having, like, you know, diverse backgrounds in this, like, you know, work? I think it will be helpful in the long term if everyone is, like, you know, agreeing with something like that. Anyone's thought on something like that?
Well, so there is like a white paper, but yeah, I mean, that's still like 70 pages long. I don't know how many people actually read that. And there's some, you know, perspectives articles that are being published by Aviv or Sarah.
But I'm not talking about articles. I'm thinking, how could you explain like high school students about, like, you know, single cell genomics and what's going to be, or human atlas, like, you know, why it is important, why they should care about it, why should become the donors of future?
I think that's an excellent point, yeah. So a way of communicating to the public about the benefits of human cell atlas, but also I think it's culturally also much more difficult to reach out to, say, people in getting donors in Japan as well. So being able to communicate clearly in their language and their level will be very important. So yeah, I think we have a lot of homework to do. In fact, like you said, we need leaders. We need someone to take the initiative and be able to drive this, not only in your country, but also in the region. And again, I think, in contrary to US and Europe, we need to make even stronger an extra effort to come together, because, you know, we're separate countries, but, you know.
We need, I mean, yes, we can theoretically do this, but it's a lot of work, and we probably need substantial support More than one or two person that really have to be. It's a lot of work, with the domestic coordination. It takes one person, half or 82 person. Yeah. I see.
Yeah. So it's domestic coordination and regional coordination. Yeah.
We are learning from the coordination in Japan that it takes more than one person to coordinate within a country with many things. And so if you want to coordinate internationally, you probably need a couple of more people full-time as a scientific coordinator of this kind of interaction.
And in a way, I think we need that because there's no correct. Yeah, if you want to unify the region, we do need a unifying group that is willing to work with different countries in Asia.
I fully agree on the point Pierre made. Wanted to bring Miguel into the conversation. Miguel? So wanted to get your thoughts. How do you address the issues that people have been discussing here? One is, what is the purpose of the genetics you're doing? One is, how do you get access to the tissues that you need? And the third is the rules and harmonization and so on.
So first of all, apologies. I've been getting lots of messages right now. So I think we should not be obsessed with rules because that can create problems. Breakthroughs come out of breaking the rules. I think at this stage of things, that many things are unknown, what we should do is be flexible and understandable of different ways of looking at things and let things evolve progressively so that in the end those rules become natural. That's one consideration. I don't think we should also be very obsessed with having tissues collecting from every countries, that they go to some specific banks or things. I think every country can do the things the way they prefer. And then in the end, I think it comes to us, the individuals, to make the effort to interact constantly to make those networks be really, really strong. And then when there's not just one leader, but it's all of us are leading the thing, is when actually we can have big breakthroughs that ultimately lead to very useful rules. That's my opinion.
Well, perhaps you can say, what tissues do you have access to? And
we are trying to work with clinicians from all over China. First, we're focusing on the region or the South China region. So we have access to almost every tissue. The conditions in which those tissues come.
Post-mortem or?
Post-mortem, also from biopsies, also from biopsies because of medicine is evolving more into taking biopsies from different organs. I'll give you an example. So amyloidosis is an important disease, amyloidosis of the heart and other tissues. So it's becoming more and more popular to do biopsies of organs, even as the heart, that you could think that you take a biopsy, you could create a big problem. So I think medicine is becoming, is going more towards that direction. So in China, and probably in any country, that you can do that. Those things are very experimental, so there's no exact protocols to be shared by everybody. I think as we move, we will be seeing what is the best way to conserve those tissues. And we're creating very many new technologies, like these spatial, these stereomics that I was speaking the other day. We're still not sure what is the best way to process those samples. We're going to be processing them in many different ways. We're going to be working with people that will tell us how they prefer to process. And then when we create, together with these people, these large databases in which we compare them with all these other individual single cells, then we'll perhaps be a little bit more aware of what is the way of doing things. But I believe that all these goals of how to do many things is perhaps the final stage of the the Human Cell Atlas 1.0, so that then we can start doing things, with a much higher level of complexity, with a much higher level of regulations. Of course, ethics regulations should be, very strict from right now, but all the type of rules that can guide us. So, in China, we can have access to almost anything, and of course, everything in an ethical way. I don't know what some people are doing in some places in China or outside China, but, the Chinese Academy of Sciences and the China National Bank, we follow the most strict regulations worldwide. The data sharing, that's an interesting thing. So I guess that every country has its own regulations. China is very careful with genetic information of Chinese patients. But this means that we cannot deposit the raw data in repositories abroad, but you can access the data. So we can create a link for you to work with any data of us. You can come to the China National Bank, see the raw data yourself, analyze everything, and then, but in order to play with the data to do analysis, you can only do with filter data. But you can help us see what those filters need to be. And then I think that's not a big problem. Many people ask us this question. I don't think that's a problem. And that's just for Chinese tissue. We can sequence tissue from anywhere else in the world, and then that one, you can put the data where you want.
So just to clarify, the data has to be hosted in China, but can it be downloaded in Singapore? Can we download the data?
Not the raw data.
Not the raw data.
If they are from Chinese nationals. But you can have the filter data, filtered by the highest standards that you may think of.
Filtered meaning?
Handled in a way that you can process them. So variation and but everything. I'm not in the pipelines of analysis. We have like 50 different people at least doing those things. So probably they would be better in terms of telling you the details of how this filtering happens. But I mean, it's done like it's done anywhere else for processing data, obviously. So we have a long history of genetic breakthroughs. So I think that tells you that everything is done following the highest standards possible.